All right, and part two, here we go. So we're at chapter 10, section 3, which is all about DNA replication, which admittedly is tough to kind of explain without drawing it and showing it to you. But what I strongly suggest is that you go to my website, mrhbio302.weebly.com, go to the book, right? And there are pictures that can help you understand what's going on as I explain it. So, A, watch this video because this will help you. So I'll pause. Hopefully you watched it. Now, nucleotides are like letters in the alphabet. In different orders, they produce a very large variety. Basically, when you have letters and you put them together, they make a word. When you put words together, it makes a sentence. When you put a sentence together or sentences together, they make paragraphs, which make books. And then when you put a bunch of books together, you have what's called a library. So now if I was going to relate it to the nucleotides are individual A, T, G, C, and U's, because we're looking at RNA actually. A codon is a group of 3 nucleotides that code for a certain amino acid. So 3 letter combinations make up one of the 20 nucleotides. When you put a bunch of codons together, they become genes. A bunch of genes in one area become chromosomes. And when you put all of the chromosomes in a body, that becomes a genome. Okay? So I know this might sound a little confusing, but there are 20 amino acids. No, I do not expect you to know these off the top of your head, but I do expect that you can recognize the abbreviation and put it with an amino acid, okay? So instead of words, nucleotides will tell the correct order of amino acids. The amino acids are put together to build proteins, okay? So proteins are made of amino acids. Proteins are the polymers. Amino acids are the monomers. These proteins perform functions in our body and cause reactions to occur. You know that enzymes are a protein. Each nitrogenous base only pairs with one other nitrogenous base. Double dating, everyone has cooties. A always goes with T, G always goes with C. The base pairs are held together by a hydrogen bond, which is in chapter 10, section 2. We know Antoine Chargas rule. A, the number of A will be number to the equal to the number of T's, or the percentage of A's is also equal to the percentage of T's. Same concept with C's and G's. Now, this is important to know. The leading strand is the one that goes 5' to 3', and the lagging strand is the one that goes 3' to 5'. But they are complementary to each other. If you know one side of DNA, you can always figure out the other half of DNA. These are complementary base pairs and are complementary strands. So, what is the difference between the bonds in the backbone of DNA and the bonds between nucleotides? There's a question you should be able to answer. Now, when we talk about DNA replication, I know this is going to seem confusing, but I want everyone to think real quick. This is what DNA looks like undisturbed. Double helix, base pairs all in the middle. Then there's an enzyme. How do I know it's an enzyme? Because it ends in what? A-S-E. So an enzyme comes along and literally unzips, unwinds the DNA, breaking the leading strand and the lagging strand. Okay? So here's the process of DNA replication. It is the process by which DNA is copied in a cell before a cell divides by mitosis, meiosis, or binary fission from chapter 8. Once again, DNA replication is important. If you didn't watch the video before, I will pause again. Watch this video. So, I'll put this in a Google form, okay? You should be able to answer these questions. So let's just take a look here. DNA is in each of our cells, and before mitosis and meiosis, we know that DNA is replicated, right? Because we know that chromatin condenses into chromosomes. DNA is going to unzip into 2 strands. New nucleotides will attach to each half strand using the complementary base pair and the rules. A's always go with T, C always goes with G. So this is the simple version of what happens here. You have your leading strand, which is 5' to 3', and you have your lagging strand, which is 3' to 5'. Just make sure you understand that much so far. Each half works like a template to make 2 new and identical strands of DNA. Replication can occur at any spot on DNA and go in both directions. So, just so we understand that, it could happen here, it could happen further along down the line, it could happen here or further along down the line. Now, here is where replication is occurring. This is DNA that's undisturbed so far, but this DNA, which has been split apart, is where replication is occurring. This is called the replication fork. Think about it, it kind of looks like a fancy fork. These are vocab words I'm not going to trouble you with knowing each and every one of these, but you definitely want to know what helicase does. You definitely want to know what DNA polymerase does. You definitely want to know what primase does. And you definitely want to know what ligase does. Okay, so make sure you get familiar with those. Now, helicase breaks hydrogen bonds, which causes the DNA double helix to unwind and separate. Primase adds a primer, basically like it is the starting line for where replication is going to happen. So it adds an RNA primer to the leading strand, and it's going to add a bunch of primers to the lagging strand. Polymerase is going to add complementary base pairs to the 3' end of RNA primer. Again, once on the leading strand and many times on the lagging strand. We call those many times on the lagging strand called Okazaki fragments. DNA polymerase replaces RNA primer with DNA nucleotides. And then at the very end of this whole process, ligase joins Okazaki fragments together on the lagging strand. So we basically just walk through those steps. But simple. Helicase, an enzyme, unzips DNA by breaking the hydrogen bonds that hold the bases together apart. Each strand, the lagging and the leading, is a template so that a new strand can be made, like a new complementary strand can be made. An enzyme called DNA polymerase helps to bring nucleotides to the strand and proofread. DNA polymerase proofreads for mistakes. You definitely want to get familiar with this, right? This is where I would actually have you draw the process of DNA replication. But here's a summary of it. Helicase unwinds the parental double helix. So here's your helicase. And when we say unwind, right, we mean we break apart the DNA double helix into 2 separate strands. Single-stranded binding proteins stabilize the unwound DNA. Again, more details you don't need to know exactly. But the leading strand is always going to go in the 5' to 3'. So find the 5', then follow it all the way to the 3'. The lagging strand, the leading strand is synthesized continuously. That means without any breaks. So you're going to find a one continuous copy of DNA going the 5' to 3' direction off of the leading strand, which is always found going in the 5' to 3'. But look at the direction of the arrow. If the 3' is here, the 5' must be going the exact opposite way. 4. The lagging strand is synthesized discontinuously, meaning look at this 3' at this side and the 5' on this side. This is your lagging strand. And now look, here's an RNA primer. Here's another RNA primer. And here are the Okazaki fragments. Remember, these happen discontinuously. Okay? And they go from, look at this. If this is the 3' here, that must mean that at the end, this is your 5'. And at the very end of the process, after the RNA primer is replaced by DNA, DNA ligase joins the Okazaki fragment to the growing strand. So eventually what happens? All of this will be broken apart, and on one side you'll have a brand new template, or not a brand new template, but a copy, one long strand of a copy. And on the other side you will have a bunch of segments that are going to eventually be joined together by ligase. Now, DNA replication can be found as semi-conservative. Here's what that means. One strand of the parent double helix, so here's our parent double helix, is conserved in each new DNA molecule. So here was the parent, and here are the parent strands. But what do you notice? Here's the new strand. It's complementary. It's semi-conservative. Same thing happened here. We had one strand, and now we have a new copy, but we have original strands, semi-conservative. Now, what if there's an error? In DNA replication, it is very accurate. There's only one error out of every 1 billion nucleotide pairs added. Think about that for a second. That is superbly efficient. Without DNA polymerase, this enzyme is what ensures the accuracy of replication by proofreading DNA for errors, limits, mutations, and again, errors. So, make sure you understand this. Okay.